Welcome to Med in 5 channel. This video will discuss the beta oxidation of fatty acids. Human body burn fatty acids and provide energy. I will use the abbreviation Fa for fatty acids and W for the energy. There are 3 ways our body can burn fatty acids: alpha oxidation, beta oxidation, and omega oxidation. This depends on carbon position. Here we have a long-chain fatty acid. The highlight in purple shows the functional group. The first carbon, near to the functional group, is alpha carbon. The second carbon near to the functional group is beta carbon. And the last one is omega carbon. This is a simple formula for a fatty acid. There are some keywords about the beta oxidation. Beta oxidation occurs in each tissue of the body except the brain, adrenal gland, and red blood cells. The organelle participated in the process is the mitochondrion. The fatty acid need to get activated before it undergoes beta oxidation cycles. Beta oxidation will remove 2 carbons from the fatty acid, in the form of acetyl-CoA. Well, the first step is the activization of the fatty acid. The fatty acid will interact with coenzyme A, and will charge adenosine 3-phosphate macroergic compound. The product will be acyl-CoA, with a specific number of carbons. After the activization, the acyl-CoA will be transported toward the mitochondrion, to undergo oxidation. The process occurs in cytoplasm. It charges 2 macroergic compounds. Cytoplasmic activization occurs only for exogenous fatty acids. Notice the difference: if the fatty acid is produced inside the matrix of the cell, its activization will occur directly in the mitochondrion. In this case, guanosine 3-phosphate will be charged, instead of adenosine 3-phosphate. The second step includes the beta oxidation reaction cycles. The activated fatty acid, acyl-CoA, enters the reaction and with the help of Fad coenzyme and a dehydrogenase enzyme, provides a double bond at the beta carbon position. The substance will be delta, inyl-CoA. Afterward, a water molecule will enter the next reaction, affected from the hydratase enzyme, with the result of adding a hydroxy group in the double bond, beta carbon position. The product is named hydroxyacyl-CoA. The hydroxy group will help the NAD coenzyme and the dehydrogenase enzyme to form a keto group. The product will be a ketoacyl-CoA. The last reaction will be initiated from lyase enzyme. Lyase will divide the ketoacyl into acyl-CoA, which now has 2 carbon less, and an acetyl-CoA. The first cycle of beta oxidation has finished now. Acetyl-CoA will enter the Krebs cycle for energy gain, while the new acyl-CoA will re-enter the beta oxidation cycle. And the cycle will repeat. In the last cycle will be an acyl-CoA with 4 carbons and will be produced 2 acetyl-CoA. If you want to calculate the energy produced, let's take an example. Palmitic acid is a saturated fatty acid. It has 16 carbons, which is an even number. In the first cycle of beta oxidation will be formed 1 molecule of acetyl-CoA. The last cycle will produce 2 molecules of acetyl-CoA. Remain 5 other cycles, which will produce 5 other molecules. So, in total we have 8 molecules of acetyl-CoA. The number of cycles is 7, as a result will be formed 7 hydrogenated NAD and Fad coenzymes. Each of them is converted in energy. Notice the calculation. There are in total 131 macroergic compounds produced. But, since the activization charges 2 macroergic compound, the final result will decrease to 129. Take note. For the odd number saturated fatty acids, or unsaturated fatty acids with 1 or more double bonds, the pathway and calculations as well will be different. That was all from Med in 5 team. Thanks for watching. For other videos like this, please click subscribe.